said that these coordinates are relative to this. This is a clade 2 reference, okay? So the IMAP kit that you get from Illumina, okay? It's like the COVID-seq kit. Remember, COVID-seq, it was everything built in one. COVID-seq came with primers and the other reagents that you needed all together. IMAP, which African CDC is going to be distributing for Mpox virus, is the same. It comes with built-in primers. And the primers that it comes with is Yale version 1.0.0 primers, okay? So these, I'm now talking about the primers in terms of physical laboratory primers that you're going to use in your lab for the sequencing, okay? But the if you are sequencing the Mpox that you are picking up is almost certainly going to be Mpox clade 1, right? So what they've done, the people on the analysis side, is they've translated these positions, okay? But now they've translated them into the coordinates that matches the reference sequence for clade 1 instead of clade 2, okay? So if you get the Yale, and you see it's called Yale version 1, clade 1, then it uses a reference sequence called KJ642613, okay? So it's what this just means is it changes the positions of the primers around. What doesn't change, it's still Yale version 1, okay? So let's just wait for people to do this plug thing. I like how colorful the plugs are. No, let you guys just do this thing. People need your power. What? What's this bit? You know, in South Africa, when we see this, we say, Amandla! Aware, power to the people. Let me know when I can carry on. You guys have power? Yeah. Okay. Okay. So as I was saying, if we see that it's Yale version 1, okay, the reference can be, and Yale version 1 means that the primers, the actual amplification primers that you use, they're always the same. But when you're doing the analysis, then you are going to be analyzing in terms of clade 1 or analyzing in terms of clade 2, right? Because what you're amplifying, you're going to amplify the same stuff because it's the same Yale version 1 primers. Okay? Now, immediately that I've said this, why who over here during the COVID pandemic had to work with updated primer schemes? Anybody here need to update, yeah? When you were sequencing SARS-CoV-2, when you started, if you started in 2020, then you were probably using version 3 primers. Then when Delta came around, if people still remember the Delta variant, version 3 primers, they didn't work anymore. They didn't work very well, right? And then you had to use version 4 primers, okay? And then when Omicron came around, you had to update to version 4.1. I don't know if anybody here did that. The same thing, Midnight got some updates as well, okay? Why we had to do those updates is because the virus mutates and the primers no longer match the virus, which means they don't amplify properly, right? Yeah. We get what's called primer dropout, okay? Now, if I said earlier that clade 1 Mpox virus and clade 2 Mpox virus, they're quite different. Do you think that we can use the same primers for clade 1 and clade 2? Can we use the same primers for clade 1 and clade 2? It's a question. No. Yes? The answer, this is the correct answer. No. Stephen, go ahead. What? Yes. How different are they? So they are at least hundreds of base pairs different. If not, I think it might even be 1,000 if I remember correctly.
Because now my other thinking was much as I have a point would be that in the region these primers are given is the difference. With the difference within
Great question. So the question is, does the difference matter? Is it a difference that matters? Okay. And what we mean by that is we look at where the primers are matching the sequence and we say, is the virus different in that place? Okay. And the answer is that, so this is, I can't remember exactly how many primers there are, but there's a few hundred primers. Okay. We find that using the existing Yale version 1 primers, there is difference in a few dozen places. There are significant differences. And preliminary results suggest that about 19 different amplification tiles, amplicons, the virus is not amplified properly by these primers. But if we have to wait for the new primers to be designed, we can't. We have to work with what we've got. And the analysis, so in other words, you don't get a perfect result, but at least you get a result that's good enough, and it's good enough to detect the transmission that we're interested in detecting, right? Okay. So before I go onto this thing, let me actually get into this thing, okay? So the basic technique. So what we're going to do, we start with this, okay? These are reads of DNA, okay? Which format do we get reads in? What's the format that we get reads from our sequencer in? The file format. FASTQ, right? The fast, which is the quality and the sequence, right? Then we map to a reference, right? So we find the position of the reads on the reference, okay? What's missing from this description, I'll show you when we do the actual analysis. The first thing we actually do is we filter away the human. So we make sure that we don't have any human data left to confuse the analysis, okay? And we remove the sequencing adapters. That's all gone. And then we map to reference. And here we obviously have to choose the correct reference, clade 1 or clade 2 reference, okay? And this might evolve because, like, some people are using a clade, like the B.1 reference from the global clade 2 outbreak is even different from the main clade 2 reference, okay? But for now, we've got a single reference. We basically have one reference that people seem to be using at the moment for clade 1. But it's everything, that's why I said to Sophonius, it's difficult to do this teaching because the whole world is changing. People are designing new primers, they're using new references. So that's why I'm emphasizing this thing, because I want you guys to be very aware of these things, because I know you mostly are coming from a lab background, and when you're doing this analysis, you must know when we change the reference or those kind of things, what does it mean? When we change the primers, what does it mean? And then the colorful things here are your inferred, are the differences to reference, right? And then the next thing that we do is we trim the primers, okay? So we've identified where, and here is one amplicon, and there's another amplicon, and there's a third amplicon, right? So these are the three amplicons, and we trim the primers from the reads. Just going back one picture. Why are we trimming the primers sequence from the reads? Does anybody know why we trim the primers? No, not you, Stephen. No. What are we sequencing? We're sequencing what is an amplicon, the read, okay? We're sequencing, remember what's going on the screen. The primer was, the DNA was binding here and it was extending from the primer sequence, right? When we were doing PCR, right? Which means that the pieces in gray are matching the primer sequence. Which means that if there's a mutation inside of the this section, okay, where it's gray, if it's in the primer, then it's going to be this sequence will not show us the real sequence of the virus. It will show us of the sequence of the primers that we put into the PCR reaction. Okay, so that's why we have to remove this piece of the sequence. Okay, and now we don't lose data because everything is overlapping. Okay, so even though we're taking that one out of this read, those reads are overlapping with this thing. So that's why it's an overlapping primer system, tiled primers. Okay. Then we trim these primers, and for that, to do the primer trimming, we need to have that bed file that tells us where the primers are, okay? And then we call variants. So this is mentioned with Madaka. I should have changed the slides. Then we actually, from here, we look at this, and we see what evidence do we have for variation. Okay, so if we look there, look at that red. It's red. Everything is saying red there, right? So we think there's a red variant there, okay? Over here, some yellow, some blue. So now we've got a problem here. Here it's red again, okay? Here, almost all yellow. So over here, we say that this is a red variant. I don't know why that one didn't call a variant. There should be a variant down here. But here, things are complicated. It's a bit of yellow, a bit of blue, which means that we're not sure what the variant is here. Okay. Now, two things can cause this kind of mixture. Okay. The one thing that can cause a mixture is if somebody is infected with two different strains of the virus, right? Co-infection. Then they can have maybe 20% or 30% of the viruses has a C in that position. Maybe 70% has a G in that position. So now we don't know if it's a C or a G. Okay? So that, the number of, so we look at the depth here, right? To understand the variance, we look at the depth and the, what's called the allele frequency, the frequency of the mutation. So here, the depth is 1, 2, 3, 4, 5, 6. Depth is 6. This is 6, okay? And its allele frequency is 100% giving us whatever the rate is. Okay, let's say it's an A, okay? Here we've got 1, 2, 3, 4, 5, 6. And we have 50% is saying yellow, 50% is saying blue, okay? So allele frequency here is 50% for two different alleles. Okay? And over there at the end, let me just try and find a gap. Yeah. But look at the one at the end. It says it's yellow. There's 3 that says yellow. So 100% frequency of yellow. Okay? But, look, this is obviously made up data. But what is my depth? The yellow variant over there, what is my depth? What is the depth of that yellow variant at the end? How many reads are supporting that position? 3, right? So in this simulated data, right? So normally what we say in our real data is that we want at least 10. We want depth at least 10, okay? let's say that I said I want depth at least 5. I don't believe the data unless there's at least 5 there. I would not make that a yellow one. I would make that gray or something like that. I would make that an N. I would say I don't know what is the sequence at that. Wherever the depth is below 5, I would say make it N, because that means I don't know what is the depth there. Okay? So, so here's the depth map. And we see here where they were using 5 as a cutoff. And we see that where the depth map is below the 5, it's colored in red as a 3, right? And so then we say that we've only got as much genome as where we've got at least depth of 10, okay? So when you were doing your COVID-19 sequencing, you would not get 100% of the genome back, right? You would get, depending on what the CT value was, 80%, 90%, 95%. If you had a bad sample, you'd get 60%, you know, and the rest would be like missing because we don't have enough data there. It's the same thing with Mpox, except it's worse, okay? It's worse because of the repetitive regions. And I'll just go back to a previous slide. It's worse because of these things, okay? When you have repetitive DNA, it means that some patterns of DNA are repeated. So now if you get a fragment, you get your read, right? Now it doesn't know whether to put the read with the one copy of the repetitive DNA or the other copy of the repetitive DNA, right? So that means that in the regions where there's repetitive DNA, we can't trust our results. We have to discard those regions when we're doing the analysis, especially with the Illumina, which is short reads, okay? So that means that we mask out, besides masking by depth, we have to mask out all of the regions which are repetitive because there will be low confidence because for us to, we can't trust the results there, okay? So we get a lot less than 100% per Mpox virus genome, okay? In my experiments, if we get 85%, it's actually not bad for Mpox. You know, whereas 85% was not nice for COVID-19, now 85% is actually quite good. And and of course, the amount of genome you get back is generally related to the CT value of the sample. So if you've got a CT, so if you're doing a PCR, your CT value is 35. Then you're almost certainly not going to get CT 35. You're going to get very little data. Okay? Yes.
On that part of the city values.
Speak louder.
On that part of the city values, I think from our lab we have got an experience. Because at one point we wanted to maybe give a cut-off at which, but you find that there are some samples that have less city values. Or rather, there are some samples that had higher city values than some samples, but yet you recover something from one that had higher city value and yet one that had a lower city value.
That's interesting. In general, the results that I've seen are still consistent that higher CT value means lower coverage. Still, in general, there can be some exceptions, but the trend should always be towards. So when you were sequencing SARS-CoV-2, did you not put a cutoff? How did you choose your samples? Did you have a cutoff for?
Yeah, we had a cutoff because of that.
30.
Sometimes depending on the situation,
Yeah.
you just sequence, but you really expect that.
There were some cases where it's an emergency and we're very interested in this case, so we're going to sequence that, right, no matter what. But in general, most people were saying if your CT value is higher than 30, then don't sequence. I'm going to use the plug just now. I think leave it open. But the other thing that's making things more complicated I don't know if anybody read the Eurosurveillance paper from the people who did the South Kivu sequencing, but they found that some of the mutations in the virus are affecting some of the PCR tests. So some of the PCR tests might not be detecting some of the clade 1s correctly. But that's another discussion, okay? But just before I get off this thing, I want to talk about one more thing. So I said that this could be caused by a mixed infection, okay? What is the more likely cause to have a mixed signal here? More likely than mixed infection. What is the most likely reason to see a mixture of maybe 20% one allele, 80% the other allele? Yeah. What? Something much more human. Lab contamination. If you're sequencing lots of samples and then you find one where there's a whole lot of where there's a whole lot of what I call is bimodal. So in other words, some of the variants are 80%, some are 20%. Okay? Then I more often than not, I worry that you are doing a batch and you had some DNA left over from the previous run or something like that, and maybe you didn't, maybe the fume hood wasn't cleaned quite well enough, or something like that has happened. Okay, so looking at allele frequencies, mixed variants, is another way to check whether your laboratory processes are running totally right. Okay, just two more things I want to talk about. Yes.
But if somebody uses negative control.
Negative control. But what if somebody doesn't use it? Then they're really playing with fire, right?
Yeah.
Does everybody use a negative control in their sequencing? I hope so, right? One of your samples must be water, nucleus-free water, right? You know what I'm talking about with the negative control, right? Okay. What's that in French? Control negative. I thought so. I thought I did it. Because this is a way to pick up contamination, okay? I mean, it's, and we saw a lot of this. We saw people claiming that they detected early Omicron before it was publicly announced. And most of the data I saw for that suggested to me it was a fault of contamination. Recombination was mentioned. For Mpox virus, it is important. Okay, Mpox virus, over a long time, not over the short time we've been seeing, but over a long time, is expanding and contracting its gene complement. It's got this kind of they call it like an accordion. You know the accordion that you play like this, okay? Because the virus, it can add and remove genes a lot through its evolutionary history, right? So if we look at one of the differences between clade 1 and clade 2, in fact, clade 1b and clade 1a, there's a gene that is just missing. Clade 1b, I think it is, I can't remember the name of the gene, but one of the reasons that they think that clade 2 might be less severe than clade 1, and even clade 1b might be less severe than clade 1a. I'm saying might be because the research is ongoing, is that there's a particular gene that's missing, totally missing from clade 1b and clade 2, okay? And whenever we're seeing this with SARS-CoV-2, we were not really seeing, we were seeing a few droplets of ORF8 and things at the end, but we mostly weren't seeing like genes that disappearing. But the genome is much more complex of SARS, of Mpox virus, and we are seeing these kind of things. But for now, what we're seeing is two things, right? Remember, there's two cycles happening with Mpox virus. The one cycle is whatever its natural host is, okay? Some kind of rat or something, we don't know what it is, but wherever the thing is living when it's not living in humans. The evolution in the natural host of Mpox virus is about 2 mutations per year, nucleotide mutations per year, okay? I'll put this on a slide. I'll put it somewhere. I was looking this thing up. So one, they put it in how they describe it is 1 times 10 to the minus 5 per base per year, and this is a 200,000 base genome, so about 2 per year. So we're saying that SARS-CoV-2 is 2 per month. This one is 2 per year. Okay, but remember, we're seeing multiple introductions into the human population, okay? So once it gets into the human population, then the human antiviral response, specifically the ApoBEC3 enzyme, okay? Do you pronounce it ApoBEC?
Yes, I got it.
ApoBEC3 enzyme. Who's heard about the ApoBEC3 enzyme? This is an antiviral enzyme. You've heard about it. I'm sure some people have heard about this now. Okay, this is an antiviral enzyme in mammals. It's causing more mutations. So when we see human-to-human transmission chains of Mpox virus, then we see a particular pattern of mutations because ApoBEC3, ApoBEC enzyme, it chooses particular patterns of DNA to mutate, okay, and that leaves us with a characteristic mutation pattern, and we're seeing that in human-to-human transmission, these ApoBEC associated mutations, it's about 6 per year. It's still not a lot. I suspect that if you are sequencing Mpox virus in your countries, you're mostly going to find exactly identical sequences. Naomi, are you seeing like a lot of very similar sequences in your Burundi sequencing? Mostly you're going to see exactly identical sequences. This is not mutating, this is, so in 1 year we'd expect about 12, mutations in SARS-CoV-2. We're getting about half that, even in sustained human-to-human transmission in Mpox virus, which means that,
and that's a global rate, so not even like a country level rate. So that's a substitution rate that's, so
yeah.
within country you expect even way less.
No, but the thing is that that's. But if we have sustained human-to-human within country chains, we haven't. We'll see what that looks like. But the thing is also, so much is still unknown about this virus. So, and also the money that was spent on SARS-CoV-2 very soon after its emergence meant that our knowledge rocketed up. We learned so much very fast about that. But this is, to me, a more complex virus. It has really complicated interactions with the host. And it's, pox viruses have obviously been studied for many years, but with a very small amount of money devoted to them. So that means that the knowledge, but that means that when we talk about genomic epidemiology, right, we are probably not, we're probably going to have difficulty directly inferring transmission from genomic data of Mpox virus. If one by one, I gave Mpox to Stanislas, and he gave it to, I forgot your name, Michael, and so on and so on and so on, all the way around to Davis. And we sequenced my virus, and we sequenced Davis's virus. We probably would end up with exactly the same sequence. Okay. So the signal is low. That said, there are other reasons to sequence besides inferring like transmission chains. We want to know the diversity, so the emergence of nucleides. We want to know new spillovers. So we've now got, because now that people have started looking, right? Because basically nobody was looking at Mpox 5 years ago, really. It didn't, like, it's a really, we'll put, we don't have any DRC people here, but they started, the first Mpox vaccine trial was started in 2018 in one of the more rural provinces of the DRC, okay? And at that point, it was a really minor thing, like, but now that they start looking, now, James, your recent paper showed that there's probably endemic Mpox in Sudan.
Yeah.
that we've not even been looking for. So we talk about endemic countries, but to some extent these are countries that where you've gone looking. You know, I mean, there's a joke about the drunk who's looking for his keys. Okay, so the man finds this drunk man looking for his keys under a street light, and he's looking for his keys. He's looking for his keys. So the guy goes to help him. You know, look for his keys, and they're looking for half an hour. They don't find the keys, and eventually the man says to the drunk guy, "Are you sure you lost your keys here?" And he says, "No, I lost my keys over there, but this is where the light is." So we have the same problem with genomic surveillance, that we've been looking where the light is, in other words, where there are people who've got the capacity for sequencing and all of those things. So we probably are still going to be finding new things. But yeah, but that's the basics.
I like that point. Like, even right
James,
now,
go for it.
if you think about the monkeypox outbreak, which clade is everyone talking about? 1B, right? But nobody's talking about 1A. You know, like there's 1B in South and North Kivu, but there's 1A everywhere in DRC, right?
And there's 1A probably in Cameroon, right?
Yes.
Cameroon is the only country that has both 1 and 2 that we know of.
Other than DRC. You know.
Yes,
DRC has 2?
Yeah.
Yeah.
In South Kivu.
So
They have
South
clade
Not
two.
21B.
No, but do they have clade 2?
No, they don't have it.
Oh, you mean like
I'm
clade
clade
2 and
the only country that we've looked at. But if you look at geography, who knows? Maybe CAR has as well.
exactly.
I mean, CAR guys, you know.
So
need
it's
to
true what
work.
you're saying. Like people look at where the light is.
But that's why what Tracy, I don't have to call it Tracy. I'm used to working with a colleague Tracy, and via the end. Sorry, Kirsty. What Kirsty did is so important, okay, to collect your details, because this is le petit déjeuner. This is not the whole meal, okay? Okay? We can, what we're trying to do is get started, but the results will come after months, if not years, of analysis and trying to make sense of what you guys discover. It's not, this is not going to be like COVID. This is going to be a sustained, slower problem for the most, especially with the continued zoonotic transmission. Okay, but the other thing is that but even for the test results, if you look at the WHO results, okay, which says that most Mpox cases in the world are coming from DRC. I don't believe it.
So it's just as most sequences are coming from.
No, not sequences. Even PCR tests. Who's got?
Yes.
I mean, PCR tests are like expensive. You know.
Yeah.
I don't even believe the WHO numbers. They are just what we can pay because it's expensive. How much is a PCR test these days? I mean, when I was buying for meningitis, it was like, I think we were getting to about $10 a test.
I don't know the figures.
But what I'm saying is that you cannot afford to do testing for most of your cases. We don't have rapid tests, okay? And it looks like we probably will not have rapid tests because people I've spoken to say that rapid tests aren't working when they're trying to develop them for Mpox. But there's still R&D. Anyway, but that's the other thing. Okay, now, that's the theory. I want to show you the tool that we're going to be using for the basics of this training. Okay, let me just move it to the right screen. If I can just find it. Yeah. Anyway, questions before we move on.
Yeah, so I think.
And speak louder.
Yeah, so along that position where we have more like where we said we have made the definition of mixed infection.
Yes.
So how do you do that when you are getting that consensus? Do you
For-
just mask it out or?
I wouldn't. What I normally do, I'll ask others to feed in as well. Normally, I set my minimum allele frequency high because I want to know the fixed positions, which are going to really help me with my genomic epidemiology. Yes, within host dynamics are interesting, but I normally say I want at least 70%, 75% of the reads supporting something, because for my work. I don't know if other people have other approaches.
No, I think that sounds good.
So the thing is that the specific analysis of within host dynamics, I feel like it's its own special, almost research problem. But I normally want to know the fixed positions in terms of evolutionary sense. Okay.
And unless you're really, really interested in
Co-infection.
between host transmission, where you think like the minor variants are going to tell you something, then you might want to look for this. But if, because then again, I often say with genomics, there is the public health questions that you need to answer. And then you always have the research questions. So
Yeah.
often we are interested in the public health, then, the research questions can come in.
So I mean, but ideally you've got a partnership between the researchers where you've got a PhD student with lots of time to devote to something, and then the public health where your boss is chasing you for the results tomorrow. Okay. And I mean I don't know. I think we still have lots of questions about like co-infection, because if we look at the dynamics of human to human, right? But especially so if we look at like the dynamics, right? So firstly, the first thing is that I actually want to shout at a lot of people, probably not the people in this room, but as a community, as a public health community, we were blinded by our own prejudices on this continent, because we were not looking for sexual transmission, and we were pretending that men who have sex with men don't exist in our cultures. Okay?
True.
So we were blinding ourselves. So if you want to take a fork and stick it in your eye so that you don't see, that's fine, but then please don't bring that to public health. And we didn't look for this thing, and we pretended that the sexual transmission was a Western problem. Okay? But the other transmission dynamic that we're seeing is in the refugee camps, in the dense settlements. And we don't know how much reinfection there is. But even on the sexual side, we don't know how much reinfection there is. We should have learned from HIV and from gonorrhea and from all those other things that sexual transmission of disease is common, especially in our mobile populations, truck drivers and lower and like people working away from home, the miners, all of this stuff, we've seen it before.
I mean, we saw it with Ebola, no? Right?
We've seen it with so many things. But anyway, so I'm just saying that as public health people, we need to put our prejudices aside and we need to look for those things. But then I'm worried that there could be reinfection and recombination and things happening, but that is more than we can normally handle from a public health lab perspective. Okay? Anyway, any more questions on this basic theory before I go into just illustrating the environment we're going to use for analysis? Have I offended everybody with my little rant? No, I have not. No? Okay. So you're all understanding this thing. Now to find... Okay. Now to find my mouse, which has disappeared somewhere. No, my mouse is just on the screen somewhere, but I can't find it.
It's at the bottom right-hand corner of the screen.
Bottom right-hand corner. No, it's not that I don't have a mouse. Just I can't see where my mouse is. There.
There it is.
There it is. Okay, I just wanted to show you something before lunchtime because we've got a lot of software to set up and things like that after lunch. So, most of you have got this screen installed, right? The Epi2Me desktop, right? And amongst the trainers, we had a lot of discussion about how should we analyze this data, what should we use, okay? And Sophonius gave me the mandate that people must be able to analyze it at their institutions without having to go onto the internet and use other countries' computers and all of those kind of things. Okay, and we know that not everybody's internet is great, so then we came up with this. This software is a bit more complicated to set up. Okay, it's from the Oxford Nanopore company, but it has the advantage that it runs the pipelines on your computer. Okay, and if we look at this, what it runs, I'm just going to look into one of the runs that I had earlier. Okay, then it runs all these different tasks. Now I mentioned, so because of the way that it's formatted, you can't read it very nicely. You can see more detail in the logs, which I'll show later. But the first thing that it does is it removes any trace of human reads from the data. Okay, so it performs host filtering. Okay, the next thing that it does is it trims. It uses a tool called Trim Galore to remove sequencing adapters from the data in case any of those are left over when you've got your data at this point. Then it maps to the reference, and then it removes, remember that thing about the gray, it trims the primers, and then it computes the consensus sequence, okay? So what it does is it finds variation in the genome compared to the reference, and it takes a copy of the reference and it puts the variation into the consensus genome. Okay, so now it makes a new genome which is also as long as the reference. So if the reference is 200,000 letters, it makes a 200,000 letter genome. It puts the mutations in there, and then it also, if there's no coverage, it replaces it with like with an N character, so that there's unknown. Okay, and finally it runs a quality control tool called Squirrel. Okay, and it does all this automated. There is a workflow behind the scenes that's running this thing. It's called RTEC, MPXV, Illumina, NF. Okay, but this Epi2Me environment, it can run more than one workflow. Okay, we're just focusing on the one workflow for today. One of the advantages to getting this set up with all of your labs is that in the future, we might, if you need to do training on something else, you will already have the software set up, and we can then say we want to analyze yellow fever virus. Here's a workflow. Run it in Epi2Me. You know, you want to do Mycobacterium tuberculosis. Here's a workflow for you. You know. It's not going into the command line, but we are going to teach about command line because that is an incredibly useful skill when you to be able to work with the bash command line when you're analyzing your data. Okay, so this afternoon we're going to sit and we're going to get this software working on everybody's computers, so that tomorrow morning we're ready to actually run some analysis on everybody's computers. Okay. And then, yeah, so that's basically me, and I think it's almost lunchtime. Any questions on anything? Or is your brain already overwhelmed by lots of information? Yes, Stephen.
So from the workflow, does it also mask the requesting ingredients?
The squirrel section does that. So Squirrel does that. No, Squirrel just checks for repetitive. Okay, after we run this thing, after we run this workflow, then let me just go back to my home. Where's my... There's this tool called Squirrel, okay? And I'll show you how to run Squirrel as well. And that masks repetitive regions. And that gives you your... Once you've run Squirrel, then let me show you the... Let me just... Sorry, find my mouse again. just rerun Squirrel on my latest data. Oh, that's the mouse. Okay. Launch. Squirrel. This should just work. I'm doing a live demo. It's always dangerous to do this. Documents. Oh. For this is correct. And this is Clade 2. Yes, I want to flag that, and I want to run phylogeny, and I want to run background. This should all work. That should all work, and that should all work. Launch workflow. Now let's see if this works. My computer is a little bit weak, so sometimes things don't work. Let's check the logs. Stop with error. Why did it error? Oh, sorry, I just forgot one of the parameters. Go back to launch. Scroll. It's not unavailable. Retry. Yeah, it's fine. Launch. 2. Yes. They changed the outputs. Oh yes, must include that. Include background. Include background. Leave that. This should work this time. So who here is using Linux on their machines? Who's got Linux? And there's one Mac and the rest is Windows, right? Okay. Most of you are using Windows. Launch workflow. Should work this time because before I just clicked one thing wrong. So, holding thumbs. Please work. Now between me clicking the wrong things and my laptop not being powerful, it is a bit anxiety causing.
So if you had to, in one sentence, describe the role of Scroll.
So Scroll has, Scroll has more than one role, which is why it's difficult to describe. So Scroll, so one of the things that Scroll does is it identifies where where the results of your other pipeline could be, like maybe should be masked, where your variant cause might be incorrect. Okay, it masks them out, and then if you select the background option, it shows you your sequence in the context of other sequences that exist out there. So, and I will show you that if this thing runs successfully, it should be running. Just check the log screen. It's still busy starting. Submitted process details. Come on, Squirrel. Doesn't take that long, but my computer really is not strong for this stuff. And it gives you a very nice quality control report once it actually runs. Please don't tell me that you are doing something crazy like redownloading stuff. Just okay, it is actually running, which means that the results will come soon. Busy running IQ tree at the moment. Just my computer being slow.
Is it one of the images on the flash drive?
Yeah.
So
So the new one that I gave you is the latest version of Scroll. It's busy running IQ tree over here, which means it's computing the phylogeny. But I'm just checking the. Yeah, that's why it's taking a little bit long. And my computer is not making life easier. So you can run Squirrel without doing I'm running the Squirrel with all of the options which are. You can run it just to do the quality control and provide the masked final sequences. But I just want to show what you get when you run it with like all of the options, because the output is quite nice to look at. I mean, and I know that we're going to be talking about phylogenetics separately, like later in the week. Who's ended up leading the phylogenetics session?
If Fanny is not around, I'll do it.
Yeah, what happened to Ifanye?
discuss.
Okay, we will discuss. The problem is, you know what? Who here has studied who here has worked on human biology before? Human biology? You. Okay, great. I can blame. And it's because of people like Stephen that we haven't figured out how to clone humans yet. It would be incredibly useful for this training if we could just make more copies of the trainers and just drop them out of a vat. Is the lunch ready?
Yes.
Okay. Let's look at this result when we finished with lunch. Okay. Actually, the one thing that we can look at while we wait for that, let's look at all the results when we're back from lunch. It's easier that way. Okay. Question.
Do you need internet to run this?
No. Once it's all downloaded, you don't need any internet. And in fact, the USB sticks that we've got here, I pre-downloaded all of the software, and we put it on here. Okay. And all of the reference data and everything that we need, which means that it'll take some time to get it all set up on everybody's computers. But once you leave here, you'll be able to run this analysis on the plane if you want, without internet. So this is one of the goals of this, the way we set up the training, is to give you a zero internet option.
So then they will install those apps on their laptops, and
yes.
They will run the samples directly from their own laptops.
Yes.
And that process, how fast or slow, depending on how powerful the laptop.
Yes. Yeah. So we did have this discussion with some other people from CAR on email. So and I had this discussion with my other colleagues in Africa CDC, look, all of the labs need to move to having a bioinformatics computing capacity. Okay, what that means is at the minimum a powerful laptop. But then we have discussed with Africa CDC if you were going to invest in a server, what that would look like as well. But at the minimum, every lab should have a powerful laptop. Okay, and this is only a few $1,000s, right? No, but I mean, compared to what's the running cost of a lab? You know, if you look at reagents and things like that, you spend a few $1,000s. You spend like this. If you buy your PCR kits and all of those kind of things, whereas that laptop you use over and over and over again, and it costs like I mean, $3,000. You can get a really nice laptop, you know, and when I raised it with Africa CDC, I said, "Are you going to buy these things?" He said, "No." They were like, "Look, the countries also need to come to the party here. So we're not gonna. It's not like we're not Oprah. We're not going to give you all a laptop to take home back to your countries." But yeah, but it's but I don't know. I mean, can you find $3,000 in your budget? I think so, somewhere. Because I know that when I buy PCR kits, I easily nanopore flow cells, the Illumina flow cells, all of these things. But yeah, okay. So I think we will look at the rest of the stuff after lunch, and then we will have lunch downstairs. Any other questions before we break? No?
This room will be locked, right? So we can.
Can we leave our stuff here?
Yeah, so we'll be here for the rest of this week.
No, but is it safe to leave our
Yes,
equipment here
so
over
we
lunch?
can leave all your things here.
Tony, will this door be locked?
It won't be locked, but this is access control.
This is access
so
control
we'll just
from
close the
because to
door.
get into Africa CDC you get access control. So it might get stolen by Africa CDC
No,
people,
no,
but I doubt
no.
it. Okay.